Greetings everyone, I am Rachael anak Hansen and my metric number is 76595. In this video, our group will present the process of binary fission in bacteria. Binary fission is used in bacteria to carry out cell division. Binary fission have the same concept to the mitosis, which both processes happen in multicellular organisms such as plants and animals, but with different purpose. The first step of binary fission process in bacteria is replication of DNA. Replication is the process by which a double-stranded DNA molecule is copied to produce two identical DNA molecules. DNA replication is one of the most basic processes that occurs within a cell. Each time a cell divides, the two resulting daughter cells must contain exactly the same genetic information as the parent cell. To accomplish this, each strand of existing DNA acts as a template for replication. Replication occurs in three major steps: the openings of the double helix, separation of the DNA strands, and the priming of the template strands, and then assemble to become a new DNA segment. During separation, the two strands of the DNA double helix uncoil at a specific location called the origin. Several enzymes and proteins then work together to prepare strands for duplication. Finally, a special enzyme called DNA polymerase organizes the assembly of the new DNA strands. That's all from me. Thank you.
Hi everyone, my name is Irene, anak Lennon. My matriculation number is 77295. Today, I will be presenting the second step in the process of binary fission in bacteria. The growth of a cell is the second step in the process of binary fission in bacteria. At this point, DNA replication, or simply put, the copying of a cell's DNA, catalyzed by replication enzymes, initiates at a specific point on a chromosome called the origin of replication. Prior to replication, a small portion of the double helix is uncoiled, separating the double-stranded DNA into single strands to be replicated. This process is performed by an enzyme known as DNA helicase, which is an enzyme that is responsible in the breaking of the interactions between base pairs. As the replication continues, the bacterial replication origins move towards the opposite poles of the cell, pulling a copy of each chromosome along with them from the cell wall attachment. The cell is then continuously extended and increased in size in preparation for binary fission, adding to the separation of the newly forming chromosomes. The replication process continues to advance until the entire chromosome is copied. Once the new chromosomes clear the midpoint of the elongated cell and move to the opposite cell ends, cytokinesis then can take place, which is the division of the cytoplasm. Thank you.
Hi everyone, my name is Sabrina Beatriz and my metric number is 76679. Today, I'm going to talk about the binary fission in bacteria, and I will continue to talk about the third step and the final steps. The third step in binary fission of bacteria is the segregation of DNA. In these steps, both of the DNA and plasmid will duplicate itself. The new duplicate DNA will attach themselves to the different parts of the cells. Finally, the cells will elongate themselves and forming septum in the middle transversely as a preparation for the next division stage. This image roughly shows you the steps of the binary fission in bacteria. Next, we're going to talk about the final steps in binary fission of bacteria, which is the splitting of cells. In this final step, a new cell wall will be formed. The new cell wall will split along in the middle, which is at the septum. The cell will finally divide into two new daughter cells, each of which contain nuclear materials and other cell organelles. Finally, the cell will be prepared and to be functioned for the next phase of life. These are some bacteria that undergoes binary fission.